Good day, welcome to my oral presentation. In this project, I wish to express the protein legomoglobin from soybean in yeast Comacagaetaella bifid cells, earlier known as piquebastores, to later be purified and used in imitation meat. It was discovered that hemp proteins play a critical part in what gives meat the meat flavor, and legomoglobins share an identical chem factor, HEMB, with mammalian myeloglobins, thus giving the desired meat taste in fake meat for vegetarians. The gene LEGHBC2 will be cloned in the shuttle vector pp coli 1, which is suitable for replication and protein expression in both E. coli and Piquebastores. pp coli 1 includes an AOX promoter for Piquebastores and is followed by a T7 promoter for E. coli. It includes the selection marker C-ofusin that works for both bacteria and yeast, which keeps the vectors short, making it convenient for handling, cloning, and transformation. It also includes a double tag, which allows for sensitive and detection and rapid purification of the expressed proteins. A restriction-based cloning strategy will be used as the chosen plasmid is created for that, and it already contains restriction sites in the multiple cloning site. The chosen restriction sites are not present in the target sequence, so first the gene of interest is amplified by PCR, and the restriction sites are added with the primers. The forward primer contains the SAL1 restriction site, and the reverse primer contains the NOT1 restriction site. The PCR product and the vector are digested with NOT1 and SAL1 restriction enzymes, incubated and placed in a gel electrophoresis, where the bands of interest 3,561 base pairs for the digested vector and 448 base pairs for the PCR product are cut, and the DNA is purified by a QYA quick gel extraction kit. Finally, ligation is performed using T4 DNA ligase. To amplify the expression vector, bacterial XL1 blue E. coli cells will be transformed with the ligated plasmid. Mercaptoethanol and the ligated plasmid will be added to the supercomponent cells to later be heat pulsed and incubated in ice. After that, SOC medium is added and incubated while shaking. Finally, it's transferred into an agar plate with zeocin antibiotic and incubated overnight. The zeocin antibiotic gives a survival advantage to the correctly transfected cells, as the intended vectors contain an antibiotic resistance gene to zeocin. This means that cells not containing the vector are unlikely to survive. The vector might be in the cell, but it could still not have the gene of interest correctly inserted. To ensure that the correct vector was generated, first a PCR amplification is done using AOX5' and AOX3' primers, already provided in the vector sequence. With a toothpick, one colony is transferred into a fresh agar plate and a PCR tube. Then PCR is performed, and the initiating heating of 4 minutes and 94 degrees Celsius allows for cell disruption and exposure of the DNA for amplification. Then the PCR products are electrophoretically separated and further analyzed using Sanger sequencing, which is possible because the insert to sequence is below the 1000 base per max for Sanger. Finally, I will transform Piquebastore cells by electroporation. The same zeocin selection marker is used, and compared to the E. coli cells, it takes Piquebastores 2 days to grow. So for a final check to see if everything was transferred correctly, I will check if the protein of interest is present in the cells by immunoblotting. This is possible because the vector added a tag RGS-His6 to the protein. So first, I need to induce protein production by growing the yeast in methanol. This is because the strong alcohol oxidase promoter AOX is strictly regulated, which means protein expression is completely repressed when grown on glucose and greatly induced when grown in methanol. So to end, the protein is purified and analyzed by Western blotting. Thank you so much for watching.